Hey everyone, I want to go through our last stage of cell respiration with you, which is oxidative phosphorylation. And then I am also gonna talk about what anaerobic respiration looks like. So this video might be a little bit longer than our past couple, but this kind of tie together everything in the end for cell respiration. So here's our overview again. Remember our glycolysis step one, outside of the mitochondria. In the cytosol, we have a glucose molecule that is going to enter in. We're going to have some stripping of electrons that makes it super reactive. That glucose molecule can then break down. A net total of 2 ATP is released during this process. We also have 2 NADH molecules released, which are energy rich. We have 2 water molecules which are released over here, and we also have 2 ATP produced. Those 2 pyruvate molecules enter into the mitochondrial matrix and go through the link stage, which is pyruvate oxidation. And during that process, pyruvate will be basically prepped and ready for the citric acid cycle. So remember that pyruvate, which was in the cytosol, comes and enters into that mitochondrial matrix by a protein. In that matrix, it'll strip away some enzymes and convert NAD plus to NADH. We have this coenzyme that adds in, but basically our pyruvate is transformed into 2 acetyl-CoA molecules. And then those acetyl-CoAs will enter into the citric acid cycle over here. And we will have one turn that will yield 2 carbon dioxide molecules, 3 NADH molecules, 1 FADH molecule, and 1 ATP. Multiply that by 2 because we have 2 of our acetyl-CoA molecules going in. So we really have 4 carbon dioxide molecules, 6 NADH molecules, 2 FADH molecules, and 2 ATP that come out of there. I feel like I'm singing the 12 Days of Christmas over here when I talk about these. At any rate, we're going to have a lot of those electrons, electron carriers. They're going to move on through, right? Those are our energy-rich molecules, even though we only had 2 ATP released in these first 2 stages. Those other ones, our NADH and FADH electron carriers, are going to head on into our last step, oxidative phosphorylation. And this is the big bang, shebang of respiration, where we end up having a large amount of ATP, somewhere between 24 and 34 molecules of ATP being released. So oxidative phosphorylation actually has 2 steps. It has the electron transport chain, which we know of already, and chemiosmosis, which we also know of. And so the breakdown of that, the electron transport chain is going to occur in the mitochondria, as we saw in that diagram before, but it's going to be in the inner membrane, not in the matrix, but the inner membrane. And this is where we're going to have some oxidative phosphorylation via this chemiosmosis process. So we have a ton of ATP being produced. Remember about chemiosmosis, there's going to be hydrogen ions that are pumped across the membrane. So those, as they diffuse and go from the high concentration to low concentration through that ATP synthase molecule, that's where we have this large production and conversion of our ADP molecules to ATP molecules. And that's where we have that generation of all of these lovely ATP molecules over here in the end. So we have a kind of a 2-step process to it. Reminder about that electron transport chain. It helps process our electrons so that this reaction occurs kind of in a stepwise motion instead of it being this large, big explosion happening, right? So we have a whole combination of these different globular proteins and molecules here within the inner membrane that will allow for this reduction oxidization process to occur. So during the electron transport chain, we actually don't have any ATP that's being made. We're just kind of processing some of those electrons so that we can support the fall of the electrons through this chain. So again, it's not one big explosion, right? And then in the end, we can see where at the bottom of the electron transport chain, when it is lower energy state over here, then we can have that combination of hydrogen and oxygen together for the production of water as well. Okay. So again, here's our view of electrons as they move through the electron transport chain. A lot like what we saw with our photosynthesis electron transport chain, right? We can see them kind of falling through each of these globular proteins and then kind of exiting out in a linear sense, right? So they're dropped off also through this location when FADH enters in. So we can see that those NADH and FADH molecules, those electron energized molecules, are going to be responsible for dropping off all those electrons through the electron transport chain. All right. Let's see, over here at the end, this is one other piece I wanted to mention. We have our oxygen molecule over here. This is our final electron acceptor. So when this is not available, the electron transport of just stops. We'll have to say that. So without oxygen, we're not going to have the final electron acceptor, and therefore the electron transport chain comes to a screeching halt over here. And then no hydrogen ions can be pumped and no ATP is produced. So this is actually a very big step, even though it seems really tiny in this diagram. All right, our last little piece has to deal with chemiosmosis and that process where we have some energy coupling occur. So remember that chemiosmosis is going to be the osmosis, not really osmosis part, but yes, it is the passive transport of our chemical or hydrogen ions across that gradient, across that membrane of the inner membrane of the mitochondria. And it works as kind of this motor system, right? So it has this proton motor force where it's going to pump and push those hydrogen ions through and enter into the mitochondrial matrix. When this happens, we're going to have the process of ATP synthase, this whole molecule here, this entire pump process and a rotary process takes those electrons that kind of follow along with the gradient there, and they're going to help generate our ATP molecules. So again, here's that whole combination of our electron transport chain and chemiosmosis. So we can see our electron-rich molecules, NADH and FADH, dropping off their electrons as it follows through the transport chain. Those electrons fall down through these globular proteins in a stepwise process so that energy can be carefully, step-by-step processed. They will come and those electrons will couple with oxygen and our hydrogen from our organic molecules and release as some water. Those hydrogen ions that followed the electrons through the electron transport chain get pumped through the ATP synthase via proton motor force. And as they get pumped through, we have the coupling of ADP and our PI molecules and this generation of a large amount of ATP, which can, again, be up to like 34, 36 total molecules during this process. So it's a really key step. There are a bunch of video clips that I linked into our original notes page, so you can look for that Harvard University one. So this is my flow chart for oxidative phosphorylation. It's going to utilize chemiosmosis. We know chemiosmosis is going to generate ATP. It does that by performing redox reactions throughout the electron transport chain, which is going to allow electrons to pass through those energy levels and control that processing of electrons. And then our final electron acceptor, remember, is oxygen. Without it, the ETC doesn't work, but water gets formed at the end. Chemiosmosis also couples with that proton gradient, which we call the proton motive force for our ATP synthase molecule. That's going to drive the hydrogen ions through our ATP synthase molecule to generate a bunch of ATP. And then that ATP can go back through and go through those redox reactions through the electron transport chain. Those final pieces of the electrons that follow through that ETC cause and assist in the processing of our hydrogen ions to go through our proton motive force in ATP synthase. So these two are friends, and they help each other out for oxidative phosphorylation. And again, it's called oxidative phosphorylation because it needs oxygen. No oxygen, this doesn't happen. So here's the final overview of our entire process for cell respiration. Glycolysis occurs no matter what. We have 2 ATP that gets generated. We have glucose broken down into 2 pyruvate. We have 2 NADH molecules that will go on later into the membrane for oxidative phosphorylation. Those 2 pyruvate go to the link step where they get oxidized and they form acetyl-CoA so that those 2 acetyl-CoA molecules can enter into the citric acid cycle or the Krebs cycle. Krebs cycle turns twice. Why? Because we have 2 acetyl-CoA molecules that came from our 2 pyruvate molecules. It'll turn twice. We'll have 2 ATP that's released. We will have our 2 we'll have our 6 NADH molecules, and we'll have our 2 FADH molecules that come from there. From our pyruvate oxidation, we have 2 NADH molecules. Again, our electron acceptors over here, along with the original NADH molecules from our first round, they're all going to come over here to our oxidative phosphorylation process where they drop off their electrons through the electron transport chain, where it couples with oxygen and we have water released. Those electrons are going to pump through hydrogen ions through that ATP synthase molecule, which will then process ADP and PI molecules and create a bunch of ATP, right? So in the end, we can have around 30 ATP, but if it all depends, we could possibly have up to 36, 38 ATP, depending on the organism and the efficiency as well. So this is kind of a slightly out of date. Could be up to 38 ATP, which is incredible. Again, there's another clip that goes along with this. The last thing I want to talk about is anaerobic respiration. So if I go back to my thing over here, we know glycolysis occurs no matter what. It doesn't require oxygen, so we don't care if oxygen is present or not. This step is always going to happen. If there isn't oxygen, then the rest of these steps don't really function, right? You need oxygen in order to accept those electrons from the electron transport chain and create ATP. If it's not there, this entire mitochondrial stepwise process isn't going to happen. So instead, the cell can go through a process called anaerobic respiration, which means without oxygen. And it can still generate ATP, but it can't make that much. It's going to only make 2 ATP during anaerobic respiration, but at least there's still ATP being formed. Otherwise, the cell would suffer greatly with only a small amount being generated. So instead of having our oxygen being that final electron acceptor, There's some different types out there. We could have sulfate, nitrate, um, or just sulfur in general that can accept those electrons to help with the production of ATP. So there are some organisms that can't survive in oxygen, so they're going to perform anaerobic respiration to still get the energy they need to survive. And then there are other ones called facultative, anaerobes, which will go through anaerobic respiration when there is oxygen present, and they might switch to fermentation when there isn't. And so that's an example of us as humans when we're exercising and get to the point of like exhaustion where we're breathing really, really hard and we have very minimal to no oxygen entering in. Think of our muscle cells on that cellular level. There's very little to no oxygen entering and exchanging into our muscle cells. We can shift into fermentation process or anaerobic respiration so that there's still a little bit of ATP being made and our bodies don't just shut down. So what does that look like? Again, it's going to occur within the cytosol, just like glycolysis, because our first step of glycolysis is going to occur. Then pyruvate is produced, right? And then when no oxygen is present, it will go through fermentation and we will have some products, either ethanol, aka our alcohol, will be released depending on what type of organism. Lactate or lactic acid could also be released. And there are a couple other products. If there is oxygen present, then it goes through our citric acid cycle and our oxidative phosphorylation. Okay. Um, So again, here's our breakdown. We'll have glycolysis occur, and then whether or not oxygen is present or not will go through fermentation. Again, it'll occur in the cytosol. We don't need oxygen because that's the whole point of fermentation or anaerobic respiration is not requiring it. The ethanol production occurs for our bacteria types. Or our fungi types will release ethanol. And for us as humans, some other eukaryotic organisms will produce lactic acid and generate 2 ATP. If we do have it, we can have that full process and a large amount of ATP produced at the end, which is huge. So there's two types like I referenced. There's alcohol fermentation and there's lactic acid fermentation. We got our bacteria, our yeast, and some fungi that go through alcohol fermentation, whereas lactic acid is some fungi, some bacteria, and human cells. So when you get these, the most common example you'll see is going to be related to human cells versus yeast. So those are the two that you should be able to for sure recognize for each of these types, okay? They're both going to start with pyruvate, and then again, what gets released in the end is ethanol and carbon dioxide for our alcohol fermentation. Those of you that might have parents or older people that you know of, friends or family that are really into brewing their own beers or whatever, their wine making or their cider making. That would be an example of what happens with the yeast. And the same thing with baking. So a little bit of ethanol or alcohol gets released and carbon dioxide. Any of you that tried to do maybe some root beer making back in middle school, you know who you are. That was this process of alcohol fermentation. Um, Lactic acid fermentation, again, is commonly associated to the muscle cells, but it is used for some common things like cheese making and yogurt making, which is kind of cool. Lactic acid, oftentimes a misconception is that it's connected to a buildup in muscle cells, which causes our muscle fatigue. So when we're exercising really hard and then struggling, to breathe, and then our muscles ache really, really bad. People used to say, hey, that's because of this lactic acid fermentation, and lactic acid causes muscle fatigue and pain, it actually has to deal with some potassium pieces. So you can always look into that. But that is a really common misconception that um, is associated to it. But anyway, lactic acid gets produced during lactic acid fermentation. Go figure. So those are our two types of fermentation. And the last piece is we actually can have some alternative sources of fuel besides glucose, and I mentioned this earlier, that can go through cell respiration. As long as we have those main components of our carbon molecules in there, then we can get what we need. So we can see that happening with the fatty acids entering in to different areas of our respiration process, and we can also see that with our proteins. So it can be broken down and still utilized for energy, since we know proteins and fats also provide energy. Um, our sugars, our glucose molecules, are most commonly used because they go through all three of these main steps here, and they're used most effectively and efficiently. But know that we can break down our other macromolecules and use them too. So there you have it. That's our breakdown of oxidative phosphorylation and anaerobic respiration. Let me know if you have any questions. Have a great day.